Nice to see everyone. Thank you for all taking a little time to learn a little bit more about Zero Diagnostics. If COVID has taught us anything over this past year, it's that whole genome sequencing is really the future of molecular diagnostics for infectious diseases. But we've always believed that sequencing is not enough, that you need the right enabling technologies to be able to generate clinically actionable data rather than just research data. And that if you could do that at scale, you have the opportunity to really revolutionize and transform the way infectious diseases are done. At DZD, we've spent the last few years developing the technologies required to create a new class of sequencing-based in vitro diagnostics. And we've developed the technologies, including proprietary methods to enable whole genome sequencing directly from clinical samples, algorithms to be able to interpret that sequencing data and make it clinically actionable. And then we've also invested in building a world-class and unique database of whole genome sequences combined with their drug susceptibility profiles to address the other pandemic in infectious diseases, which is the growing threat of antimicrobial resistance. And by taking the lead in diagnosis, we will generate the stream of real-time data required to change how we track, manage, and combat infectious diseases in a way that hasn't been done before. Our company has focused on a first application, which is bloodstream infections and sepsis. And we focused on bloodstream infections not because it's the easiest thing to focus on, but because it's actually the hardest. It's the places where there is the greatest unmet clinical need, responsible for 1 in every 3 hospital deaths that occur today in the US, has a mortality rate of 10 to 40% depending on severity, and is responsible for $62 billion worth of costs in both hospital as well as post-care costs for the Medicare population alone. And the reason that there's such unmet need in sepsis is because the problem of sepsis is actually a problem of time. The life expectancy of a patient with septic shock, a very severe form of sepsis, without fast, effective antibiotic treatment is 24 hours. And that person can present with septic shock immediately as they come into the hospital. But current diagnostics take 2 to 5 days to return a result. And that's because it's based on culture. For a complete diagnosis, which includes not just species ID, but the antimicrobial susceptibility profile of the infection, you have to grow a culture. And that becomes the Achilles heel of all culture-based or antimicrobial resistance diagnostics. So today's standard of care, when they're presented with this problem, is that physicians carpet bomb the infection with broad-spectrum antibiotics. They take a hammer and sledgehammer approach to really drive down whatever the infection is. And unfortunately, in an era of growing antibiotic resistance, empiric therapy is ineffective 30% of the time because of the rise of multi-drug resistant organisms. In 20 to 50% of cases, even when it's effective, a patient suffers collateral damage to the kidney, heart, and liver, and experiences a 22% increase in mortality risk, even when it's effective at killing the infection. And because it takes multiple days to get through the therapy, the patients end up in the hospital 8 days longer at a cost of $46,000 than if they had a precision therapy at the very beginning of their course of stay. It's the exact opposite of precision medicine. What DZD delivers is a complete diagnosis directly from blood in 6 to 8 hours rather than 2 to 5 days. And by complete diagnosis, I mean not just confirmation of infection, but comprehensive agnostic species ID and comprehensive antimicrobial susceptibility profiling, enabling the physician to use a targeted treatment on day 0 of hospital admission rather than waiting for the 2nd, 3rd, or 4th day. And what makes this possible is our breakthrough technologies. We've developed a set of unprecedented sample prep technologies enabling whole genome sequencing directly from clinical samples, advanced machine learning algorithms, all built into an automated diagnostic that enables a clinician to make a determination within the first day of hospital admission. Our unprecedented sample prep takes a sample with 1 billion times more human DNA than bacterial DNA, which is the clinically relevant presentation for sepsis, and outputs sequencing data with 10 to 100 times more bacterial reads than human reads. That's a 9 to 10 log order of enrichment of bacterial DNA directly from clinical sample. And to put that in perspective, it's literally 1 million times better than any other sample prep system that we've seen, either in the academic lab space or in the commercial competitor space. And what that really gives us is the ability to derive whole genome coverage rather than work from snippets of DNA. So for the first time in a clinical setting, you're able to work from full information about the genomics of a pathogen rather than just bits and pieces or biomarkers of 16S or whatever else you want to think about for PCR and biomarker-based approaches. Once we have that whole genome sequence, we're able to run our algorithm Kenome against that sequence data. And Kenome is able to do comprehensive, accurate pathogen detection and species ID, as well as, for the first and only time of any company, comprehensive AST profiling directly from genomic data. And all of you guys know, because machine learning is so prevalent, that any machine learning algorithm is only as good as the data on which it's built. And we've spent the last 4 years painstakingly building MicromeDB, a proprietary data set that combines whole genome sequences with the phenotypic resistance profiles of thousands of pathogens in our data set. DZD is the only solution that delivers on all 4 principles required, we believe, to provide a complete solution for sepsis. We work directly from clinical samples rather than from culture, because culture takes time and fails to grow 40% of cases. We provide comprehensive agnostic species ID, so you're not blind to what somebody is infected with, even if it's not one of the 5 species that somebody else may be able to discover. We combine species ID with antibiotic susceptibility profiling, because if you don't have both pieces of information, you can't make a targeted therapy decision as a clinician. And finally, we build it into an automated system that sits in the hospital microbiology lab, so it can be performed locally with a turnaround time of hours rather than days. With our first application, we're targeting a $6 billion market for inpatient microbiology ID and AST testing. In bloodstream infections, there are 40 to 50 million blood cultures taken every year in order to try to diagnose a bloodstream infection and figure out what the infection is. We directly address all of those blood culture sample types that run through the microbiology lab. And by getting a foothold in an area of great unmet need like sepsis, we get the opportunity to work in urinary tract infections, CSF or meningitis, prosthetic joint infections, essentially become a funnel through which all samples in the microbiology lab would go. We have a razor and razor blade business model typical of IVD diagnostic. What's unusual is the opportunity for us to provide SaaS model analytic services that ride on top of the IVD. Because if you think about what our diagnostic is really doing, every time somebody runs our test, it generates a genomic data point that didn't exist before. And that genomic data point is captured in MicromeDB. And as it gets captured in there, we get the opportunity to analyze it at scale. So we can tell somebody from Mass General Hospital something they sequenced today is identical to something they sequenced yesterday, i.e. it's a hospital-acquired infection event. We can tell somebody at UCLA whether something they sequenced is a rare, unusual strain that's never been seen in the Southern California region before. And we may even get the opportunity to track and manage outbreaks and look at it through a big data lens so that we can prevent the next global pandemic. If this is of interest to you and you'd like to see what we're doing and hear a little bit more of our fundraise, come please see us at our breakout room. Thanks very much.